Today we'll be preparing fungal slide from pure culture of fungal plates. So materials needed, we'll be using a pure culture fungal plate grown on a potato dextrose agar. Then we're using inoculating needle. We're using Lactophenol cotton blue stain. We're using spirit lamp. We're using microscope slide and cover slide. Now, following are the techniques that will be employed. The inoculating needle will first be sterilized in naked flame. Then the needle will be used to cut a small portion and cool. Then today we'll be preparing fungal slide from pure culture fungal plate. So these are the materials that we'll be using. We'll be using a pure culture fungal plate that was grown from potato dextrose agar. We'll be using inoculating needle. We'll be using Lactophenol cotton blue stain. It's a choice stain for isolating fungi. We'll be using spirit lamp. We'll be using microscope slide and cover slide. Following are the staining techniques. First, the inoculating needle will first be sterilized in naked flame to avoid contamination. Then the needle will be used to cut after sterilizing the needle, we have to cool it gently before applying it to cut a small portion of the fungal colony. So the needle will be used to cut a small portion of the fungal colony down into the agar medium, and then to place on microscope slide. We are cutting into the agar medium because the fungi has its roots inside the agar medium. So we'll cut it down into the agar medium. Then the needle will be used to tease the fungi for even distribution. We try and separate it gently, tease it with needle on the slide. Then 2 to 3 drops of Lactophenol cotton blue stain will be applied to the specimen. Then the base of the slide will be passed through the flame for 5 to 10 seconds so that the agar can melt and it will be easier to put a cover slide. So the cover slide will be gently lowered to cover the slide from one side to avoid trapping air bubbles. Then the excess stain will be cleaned and the slide will be examined under the microscope at ×10 objective and ×40 objective. In the lab, in laminar flow. Now this is our pure culture fungal plate. These are Lactophenol cotton blue stain. These are glass slide. These are cover slip. First, we sterilize our inoculating needle in hot flame. Then we will cool it gently so that we will not kill the organism that we want to pick pure culture from. So bring the flame closer to create a sterile environment. So gently pick small fungi from pick it down into the agar. So I said earlier that we'll cut small portion of the fungal colony. Then we put it on the glass slide. Then we tease it. We try and break into smaller pieces. So we'll have even distribution of this fungi. Tease it. Then we flame an inoculating loop again, not to contaminate the environment where the work is being done. Then we leave the inoculating loop on one side. So cover the place. So we'll take our slide and add 2 to 3 drops of lactophenol or some blue stain. We'll pass it through flame for like 5 seconds. Then we'll leave the slide and gently cover with cover slip from one side. We gently cover it with cover slip from one side to avoid trapping air bubbles. We lower it gently and press the cover slip gently. So this is our prepared slide. We examine under ×10 objective microscope and ×40 objective microscope. Now we'll pass through flame so that it will be easier for us to press the cover slip. It will have melted the agar. So it is ready for checking under the microscope. This is a fungal slide prepared from a pure culture of fungal plates. Now I want to examine under the microscope. This microscope is an optical equipment that uses light to reflect image. So we have different parts. We have the eyepiece, the objective lens, the stage, we have the base, we have the arm, we have the coarse and adjustment knob, fine adjustment knob. So first we have connected the microscope to the light source. So we on the light. Then we use the coarse adjustment knob to bring the stage down. So it will be easier for us to mount our fungal slide prepared. So we make sure that what we mounted is directly under the rays of light, as you can see. So we fix our eyes in the ocular and raise the stage up gently until we have a sharp object. So after we get a very sharp object, we use fine adjustment knob to fine tune it. Then we have our well focused fungal slide. Yes, after we focus the fungal with ×10 objective, you can see some view of the fungal slide, the organism. You can see the mycelium, you can see the hyphae, the sporangium. Now, for us to see a more bigger view showing the internal structure, we move our objective lens to ×40, so it can show us a more bigger view. Yes, we can now see a more bigger view of our fungal slide. Now at this stage, we have to increase the light a bit, and then we use a fine adjustment knob to fine tune. Now you'll see the picture of this slide at the end of the video. Thank you.